After phage lambda, let us study about the next bacteriophage virus, that is phage M13. Now, what is this phage M13? This is a filamentous phage of E. coli, which infects only such cells which contain sex pili. Means, for example, this is a E. coli cell. So this particular virus is basically filamentous phage for E. coli, and this is the sex pilus of E. coli. For example, this is the sex pilus. So this virus lands on this sex pilus and delivers its DNA into the cell through this lumen. For example, this is the DNA. So this virus, and this is the virus, it inserts its DNA here, and now this DNA gets inserted in the cell or entered in the cell. This is its DNA. It has single stranded DNA. This thing is very important, that the DNA of this virus is a single stranded circular DNA molecule having about 6,407 bases. Now when it lands on the sex pilus and delivers its DNA to the host cell, the lumen, through the lumen of pilus. After delivery, this DNA gets converted into double-stranded DNA, which is known as replicative form or RF form. Now this RF form or replicative form, it again start dividing and starts doubling this double-stranded molecule. It is called replicative form, and then it forms about 50 to 100 RF molecules per cell. Means again this starts dividing. Further again it starts dividing, and like that in a cell we'll get about 50 to 100 RF molecules, means are replicative forms, this kind of double-stranded DNA. Finally, single-stranded DNA molecule is produced, means at the end again single-stranded molecule is produced, and this single-stranded DNA when it produced from the replicative form, it extrudes from the cell as M13 particle. From here it extrudes out from the cell in the form of M13 particle, and progeny molecules are packed upon extrusion through the cell wall. Means this DNA gets extrudes out from the cell. These cells are not killed by this virus, only their growth becomes slow. So this virus does not kill the cell, only the growth of the cell becomes slow. Foreign DNA of about 500 base pairs can be cloned into this multiple cloning sequence. For the development of M13 based vector, this RF replicative form, it can be purified. Means we have to purify this replicative form and manipulate it. We have to manipulate this replicative, means we have to purify this form and manipulate it. Foreign DNA inserted into M13 vectors can be procured as a single-stranded DNA. Means if we want a single-stranded foreign DNA, then we can use this replicative form of phage M13. And these clone genes are very useful. There are some techniques where these types of genes are used. One of this is DNA sequencing, and another one is site-directed mutagenesis, which we have to study in preceding sections. Thank you.